Okay, so we keep this whole series as anatomy of questions, and here we want to discuss about the detailed structure of a question, because if you understand the structure of a question very clearly, you can answer them fast. Okay, and sometimes understanding the structure is more important, even important than knowledge of the subject itself. So today we will talk about unit 1 and unit 2 questions. Okay, so 6 questions from each unit. Approximate number of questions you can expect from these units in part C in CSIR NET exam. So this is okay. So we are talking about different types of questions. So this is another part of anatomy with a question, and this is about unit 4. Okay, so let's see few of the unit 4 questions again, and what it takes to solve unit 4 questions, because unit 4 is the most diversified unit that covers cell signaling, that covers cancer biology, it covers immunology as well as host pathogen interaction. So we'll see few of the questions. Particularly in my opinion, I'll go with the cell signaling, I'll go with cancer, and I also go with host pathogen interaction. I don't generally follow immunology because the question standard I've seen in the immunology question for last few times, most of the time they are very difficult, and it's beyond comprehension for a biology student to continue. In fact, most of the questions are asked from the research papers in the modern era. If you don't know that, if you don't prepare that specific of an information, you will not be able to answer that. So my bait here will be with these 3 type of questions. I prepare all these 3 topics from unit 4 very well, so that I can attain all of them. So let's begin with the first one. You have isolated a short chain polypeptide from a bacteria which, upon administering to animal cells in culture, is demonstrated to get transported to mitochondria and to cause mitochondrial membrane disruption. This peptide is being considered for its therapeutic effects on cancer and is to be tested for its anti-cancer properties in the following models of the disease. Okay. So again, read it once, because a lot of information, sometimes using complex sentence make it difficult. You have isolated a short chain polypeptide from a bacteria which, upon administering to animal cells and culture, demonstrated to get transported into the mitochondria. So the polypeptide can go inside the mitochondria and can cause mitochondrial membrane disruption. So if a polypeptide, means a protein, can disrupt the membrane of mitochondria, that means it's going to kill the cell, because if the mitochondrial membrane is disrupted, it will release cytochrome C from mitochondria into the cytosol. Now that cytochrome C, along with APAP1 and procaspase 9, form apoptosome complex that ultimately destroys the cell. You know, like they activate the caspase 8, I believe. In this, 8 in case of extrinsic, but in this case caspase 9, and they finally can kill the cell at the end. Now the question is, this signal cascade ultimately activating caspase 3 that activates nucleases that kills the cell. Now the question is, to answer this question, we need to know this mechanism of apoptosis, which is intrinsic mechanism of apoptosis, or intrinsic mode of death. So intrinsic apoptosis involving pore formation in the mitochondria, and here they said that which of the following properties in the following models of the disease, like the therapeutic effects of cancer, and it is to be tested for its anti-cancer properties for the following disease. One is the cancer with APAF1 mutation, cancer with BAX gene mutation, cancer with BAC gene mutation. Which of the following is likely to provide the best effect to induce apoptosis? Now think about it. Among all these 3, because the protein that we have isolated has a property to kill the cell, that means the protein is tumor suppressor effect, right? They have a tumor suppressor effect. Now all these 3 genes are mentioned, APAF1, BAX and BAC. Among these 3, all this, they are involved in cell death, so they are the resultant of the tumor suppressor product. But the protein we have discovered, they disrupt the membrane of mitochondria, that leads to the release of cytochrome C. But APAF1 is not present in the mitochondria. APAF1 is present in the cytosol. It's produced in the cytosol. So our target protein here, the protein that we are working with, definitely are BAX and BAC, not APAF1. So B and C both are correct as per our opinion. So option 2 should be the correct option. Because if you recall the mechanism of the internal or internal mechanism of apoptosis, you know that this back both cause the release of cytochrome C. APAF1 present in outside of the mitochondria, it's linked, and then they activate caspase 9, caspase 9 activate caspase 3, and then finally apoptosis is caused. So this is the idea, okay. So whenever you read this first half of the question, you must know that this question is from apoptosis, intrinsic pathway of apoptosis. So if you know intrinsic pathway, we attain the question. If you don't know, you should leave the question, okay, no doubt about it. So let's move to the next one. Certain chemokines are known to suppress HIV infection, whereas pro-inflammatory cytokines are known to enhance the infection. In order to explain these findings, control and chemokine receptor knockout animals were treated with pro-inflammatory cytokines followed by HIV administration, and then infection was addressed periodically. Which one of the graphical representation given below best explains the experimental results? The answer is this is not the thing here, okay. So this is mentioned wrong here in this case. So what we can see, the control is given with this circle, the black circle, and the triangle is the treatment, okay, the treated or the experimental set. Now there are these two type of results. Result is already told, you know, in the very beginning. Certain chemokines known to suppress HIV infection, whereas pro-inflammatory cytokines. So chemokines here suppress HIV and pro-inflammatory cytokines enhance HIV. Now which of the following graph properly explain this both findings together? Now for control, if you look at here, for control, the graph will be the same. You know that at the very beginning of the onset of HIV, the graph will jump first, and then once the antibody is produced, slowly the graph will go down. This is normal thing for all the case. But the question is the comparison between a control and experimental data set. Which will give more magnitude of action, which will give less magnitude of action. Now we know that we took this chemokine receptor knockout animals, right? So this chemokine receptor knockout means the pro-inflammatory cytokines are not going to impact, because if these receptors are not present, then where the cytokines are going to bind? So there is nothing to bind. So as the cytokines cannot bind with the receptor of the chemokine, then the impact will not be that much. Means the impact of this pro-inflammatory cytokine to enhance the HIV is not possible. Okay, although the HIV can cause the infection at the onset, but that impact is not that higher than a normal situation or a controlled situation. In fact, in this case, the impact will be lower because the lower than the control. So what we can see is that among these four, where we can find out the magnitude of this HIV infection lower than a control, that is this one, okay, lower than the control. And how much lower? If you look at here, this is also lower, but the difference between these two is that in this case the value is given very low, but in this case the difference between the two is quite much. So the difference will be quite much from the wild type, from the control, okay, because if the cytokine receptors are normally present, then the pro-inflammatory cytokines are going to bind with them and they will signal them to influence the infection even more. So then you can see the spike in the experiment, but that is not the case. The chemokine receptors already taken out, knockout are produced. Knockout means that gene is not present, right? So that's why this should be the result. So this is one immunology question. They always ask this kind of immunology questions that mostly are a result of different experiments and combination of different experiments involving different mice, involving different interleukins, different interleukin receptors, cytokines, chemokine receptors, and so on. So you need to read a lot of things, and not only that, but once you prepare every single details of the immune system, but still this part will remain always a big question mark in the exam. That's why I never encourage anyone to go with the immunology, because they ask 1 to 2 questions, 2 question at max from immunology in part C. in a single year, in a single season of session of CSIR NET exam. But the question is, to prepare for that, you need to read whole immunology book. Even reading the whole immunology book will not be sufficient to answer this kind of questions. Okay, so let's move to the next one. This is another immunology question. There are like this kind of question you can still answer. You know, the topics that I mentioned in the class, if you prepare that, you can still answer this kind of question. There are three substances A, B and C. Given below are the pattern of immunological responses in the rabbits when three situations. 1. A is administered along with C, B is administered along with C, and A is conjugated with B, administered along with C. Three situations. So if you look at the option, you know, by reading till this portion, nothing gets clear. You know what we are talking about, whether we are talking about A, B, C, three substance. We don't know whether the substance are proteins or fat or carbohydrate, or I don't know. So if you look at the option A, protein, B, hapten, C, adjuvant, that means when you read the first option, you know one thing, that this question is from the protein, means the antigen hapten conjugate, as well as the role of adjuvant. So these are the three concepts. You know, one concept of antigen. There are small molecules which cannot elicit an immune response, so they need to be associated with large molecule to become immunogenic, and that example, that large molecule is known as this hapten. So it's known as hapten carrier conjugate. So the large molecule is the carrier and the hapten is the small molecule, so called the hapten carrier conjugate. We already talked about that in the class. And there is another thing called adjuvant, which we haven't talked about earlier. So let's see what these things are. If I show you this picture, an adjuvant can be more complex pharmacological, typically not conjugated, and does not induce an immune response on its even when part of the combined vaccine. So adjuvant never induces immune response. But a hapten is small, more likely a chemical. When conjugated, can induce immune response. Okay. So normally the hapten need to be associated with large carrier molecules. So this is a hapten, very small molecule, failed to elicit any immune response. Carrier molecule is huge. So let's say penicillin G is a hapten, cannot produce an immune response. Carrier molecule is serum albumin. So this together will form hapten carrier conjugate, and they can induce immune response. So once you know the properties between the antigen, which is nothing but a protein in this case, the hapten is a small protein. So protein, and you know protein can be combined with a carrier, which is a big, like carrier is a big protein also. Hapten is a small protein. Okay, it's protein or many other thing, it can be anything. But the carrier is actually the big protein. Hapten is a small one, and adjuvant is more complex, as I told you, typically not as conjugate present there. Okay, so now when you read the statements, A is administered along with C, B is administered along with C, A is conjugated with B, administered along with C. So from this third statement you can get a clear idea. A is conjugated with B, that means between A and B, one is adjuvant, one is hapten and one is the carrier. Okay, so between A and B, no one is adjuvant. C is definitely adjuvant. So from statement 3 you can clearly get that, because A and B working as a conjugate. So obviously one is the protein, one is the hapten. So C is adjuvant. Once you know C is adjuvant, if you look at the option 3 and 4 will not be the right option, because where both these options says other things. So now option 1 and 2 left. So by knowing one fact, by just reading the question, we can eliminate two options with again presence of mind. Now between option 1 and 2, which is correct? Now let's see. In the first graph, A is administered along with C. So C is adjuvant. When A administered along with C, no immune response, no such response is seen. Okay. And in second case, where B is administered along with C, then you see a response. So we see a response against B. That's something really unique. A response against B. So that means B can elicit a response there. Okay, fine. And at the end, when we have A, B both, then you see the response of B is high, the response of A is less. Okay. So now what we can see, when B is there in presence of C, there is a response, and the response is possible by the big molecules. The immune response generally caused by or induced by bigger molecule, not by the hapten only. So hapten is a molecule which is small, cannot elicit immune response on its own, right? So, and we know C is not the carrier, so obviously B is not hapten. B is not hapten. So C is adjuvant, B is not hapten, so B is carrier protein. So if B is protein, then who is hapten? The only left A is hapten. That's why A failed to elicit any immune response, even in the presence of C. So A is a hapten, B is a protein, C is adjuvant. So option 2 is the correct option. So now, if you know about this hapten carrier conjugate, and if you know about this adjuvant, you can still answer this question. So this is quite easier compared to the last one, because this is a direct flat question, although it requires you to understand the graphs, but still manageable. Manageable. Another immunology question. The relation between cellular immune response generated against hepatitis C virus is the critical determinant of the outcome of infection. Given below are the representative figures of cellular immune response in column 1 and the various outcome of infection in column 2. So from there you need to choose the correct combinations. Okay. So this is the 2-4 separation from there. Okay. So we are talking about acute infection, chronic infection, and resolution. Means the infection is already over and our immune system already is protecting us. Now here you can see, this is the infection graph. Actually the write-up is not getting clear in here. The symptom, the one with this beads, the blank beads on a string, this is for the immune response that we are going to see. Okay, this is what we will see in all these cases. So if you look at this one, A, what you can see in A is that response is very fast. So the HIV onset of infection is very fast, and then after some time it slows down and the HIV is gone from the body. And this is our immune response in the side. This is our immune response. Okay. So clearly see both the situation. Okay, the HIV and the immune response, how the immune response is working. This is the HIV titer that goes and slowly goes down. Immune response is winning against it. So what we can see, this kind of response where the agent present in the body for a short span of time and then immune response easily can defeat them, is an acute response. So A is acute. There is no doubt about it. A is acute. Okay. So A is acute, A is 1, that's fine. So let's find out which of the options carry A with 1, 2 and 4. So 1 and 3 are not correct. So between 2 and 4 now we need to find out the option. Now the second graph, if you see that the infection increased at a time and then slowly goes down. Okay, and then again after some time it come back and continues like that. So in this case the infection is not getting decreased. You can clearly see the HIV infection is not getting decreased. It is not totally resolved. So it is definitely not resolution, because in resolution you can see that ultimately the HIV infection is gone. So you'll see the end of the graph like this. So this is resolution and this is a chronic infection. So in chronic infection what happens? Infection start first rapidly, like the massive infection, then slowly goes down while the immune response start up. This is the immune response start up, it goes down and then slowly again second time can come, third time can come, like continues like that. So our immune system will also continue like that. Okay, so it will win if the immune response goes up, then only it will fall down. So till this part it's a example of chronic HIV infection. Okay, so again this question can be answered if you know the immune response. There are primary immune response and secondary immune response. If you know about immune response, how it is maintained, you can still answer this question quite easily. Okay, but still these are manageable questions from immunology. Now let's talk about some other question. As topoisomerase play an important role during replication, a large number of anti-cancer drugs have been developed that inhibit the activity of these enzymes. So if we inhibit the activity of topoisomerase, the replication fork will stall, thus the DNA synthesis will stop, thus the cell division will stop. This is our general idea. Which of the following statement is not true? Means which of the following statement is false? Whenever you find this term not true, just substitute it with false and read it with the false term. So which of the following statement is false about the topoisomerase as a potential anti-cancer drug target? Okay, so we need to find out the false statements. So first one, as cancer cells are rapidly growing cells, they usually contain higher level of topoisomerase. This is one thing. So yes, obviously they will need that. It's true. The second one now, if you look, the transient DNA breaks created by topoisomerases are usually converted to permanent breaks in the genome in the presence of topoisomerase targeted drugs. True or false? Yes, true. Because topoisomerase, if we target topoisomerase and topoisomerase is not doing their job, then the topoisomerase, then whenever there is a single-stranded DNA damage, double-stranded DNA damage, those damage of the DNA strands remain as it is because nobody is there to fix them. As cancer cells often have impaired DNA repair pathways, they are more susceptible towards topoisomerase targeted drug. That's also true because in cancers they need to divide so rapidly, so fast, they do not get the time for the impaired DNA repair pathway. To go with the continuous DNA repair pathway or conventional DNA repair pathway. So they have an impaired version of the DNA repair pathway. So they are more susceptible towards topoisomerase targeted drugs. The drugs which specifically target topoisomerase usually do not affect normal fast growing cells. Now this should be our prime criteria if we are developing a drug against the topoisomerase activity to prevent cancer, because the topoisomerase must be present in all the cells, okay, in all our normal cells as well along with the cancerous cell. So if administering that drug is going to prevent the function of topoisomerase in all the cells, then a normal cell division in our body will also stop. Then it will be a big problem. So normally what we know, even if we take the drug, that drug is going to act on the topoisomerase. But what will happen, in some case, that drug will not allow the topoisomerase to do their job. So that will result in the DNA breakage and all. Now our normal cell will engage DNA repair mechanism to fix it, and we can still fix in the normal cells. But in the cancerous cell, the DNA repair mechanisms are not that active, and the DNA repair systems, due to inactivation, the cell will continue with this problem with topoisomerase and DNA break that ultimately causes the death of the cancer cell. So this is probably our idea when we are generating a drug against this topoisomerase function. But still it will not be that fruitful, because if in a normal cell we rely on our we are relying on a normal DNA repair mechanism to repair the DNA if any damage have occurred, and that may not be repaired all the time. It's not possible. Okay, so that's also a deadline thing here. Okay, but still we can do that. Okay, so this is a question which requires your understanding of a drug target and also understanding the function of topoisomerase and the function of DNA repair mechanisms, and also a property that the cancer cells have an impaired DNA repair pathway to answer this question. If you don't know any of that, and these are cross informational questions. It's not question from a single part, it's a question from cancer biology, cell biology, molecular biology, all combined. So you can only answer them when you are good at all these things. Okay, and they are difficult because these are not combination based, only single type question. Okay, so the drugs specially target topoisomerase usually do not affect normal fast growing cells. This is a false statement that we can take, because the fast growing cells, whatever normal cells are not fast growing, but whatever normal cells we have, the normal cells are going to be killed. The normal cells are going to be, again, the same process will occur for the normal cells as well, because the drug is going to act on all the cells, normal cells as well as in the cancer cells. So in the normal cells, the topoisomerase will be also out of function, so they will not cause any release of the replication fork. So replication fork cannot move, it will stall, and the cell division for normal cell will also block. So the fourth statement is totally wrong, that this drug can targetedly impact the cancer cell. No, this drug will not targetedly impact a cancer cell. That's why we generally don't produce a drug that prevents the topoisomerase activity. So it's not going to help. Okay, remember that. Okay, so let's begin with another question. The next question now. Let's take a color. Okay. Immunoglobulins have therapeutic applications in cancer treatment, infection clearance, and targeted drug delivery. For this reason, immunoglobulins are briefly cleaved by enzyme pepsin. Following are some of the statements regarding the brief digestion of immunoglobulin by pepsin. So if you read the question, you know one thing, the topic of the question. The question is asked from the protein hydrolysis of immunoglobulins. So if you use pepsin, trypsin, chymotrypsin to the immunoglobulins, what kind of fragments we can get? And also papain sometimes used to do that. So for 4 different proteases, you need to know how exactly they are going to fragmentize the immunoglobulin, how many fragments we can get, and based on that they can give you a data set question like that, or they can give you a graphical question, or they can give you a question of pictures with loading of those lysets filled with the immunoglobulin fragments. So what we know is that if we treat the immunoglobulin with pepsin, so you need to know the result. The outcome is the immunoglobulin is fragmented into 2 different parts. One is the large fragment containing FAB2, means both the FAB portions will be present, which is near about 110 kilodalton, and rest of the FC regions are further fragmentized by the pepsin in several different locations non-specifically, and those are FC fragments. Okay, so assuming that IgG is actually fragmentized by pepsin, where the IgG has 150 kd of the molecular weight. So what we know is that what will happen here, the FAB2 fragment is generated which retains the antigen binding activity. Is it yes or no? Yes, because if FAB portion is present, this is the place that carries the antigen binding property. This is the place that has the variable region. So this will bind with the antigens. Okay, so this is true. 1 is true. So if 1 is true, 4 is not correct. Option 4 is not correct. So let's read. FAB fragment having antigen binding activity and the crystallizable FC fragments are generated. No, crystallizable FC fragments are never generated. The fragment generated on incubation with a proper antigen forms a visible precipitate. This is also truth. This is the truth, that yes, if we add antigen, this antigen can bind with this antibody and they can form a visible precipitate. We can do that, we can form that. So as it forms this visible precipitate, so 3 is also true. 1 and 3 both are true. Now look at the 4th one. The fragment generated is incapable of forming a visible precipitate on incubation with a proper antigen. So obviously if 3 is true, 4 must not be true. So 1 and 3 both are true. So the option 2 is the correct one, with 1 and 3 both present here. So this is what you know. Okay, so if you know about the use of pepsin to cleave the immunoglobulins generating the fragments, you can answer the question. They can also ask this question with the treatment of the immunoglobulins with trypsin, papain, any of that. So ultimately you need to know how many fragments are generated, because they can also ask you the question by giving you a SDS page and telling you that we load the lysate in the SDS page and we are trying to figure out the bands, and based on the bands we are trying to figure out which. So let's say this is enzyme X, this is protein hydrolytic enzyme X, this is Y. So you need to figure out whether X and Y is associated with, like, whether which one is pepsin, which one is trypsin, which one is papain. You need to figure out based on the bands you achieve, and the length of the bands will also be, like, molecular weight will also be given in this side as well. Okay, so based on that you can figure it out. So this kind of question can also be asked from the same region. So these immunological questions I attain because, you know, I haven't discussed much questions of immunology earlier. So these are the regions which you can still prepare from immunology, but most of the questions they ask are much more difficult and tricky to answer. And sometimes they also ask questions from immunology and cancer biology combined. Okay, so also practice those questions from the last few years question papers. Yes, the answer is yes. If you ask yourself this question, that can you qualify CSIR NET Life Science exam within 6 month preparation? Yes. And if you don't need to believe me, just see the results that our students are getting who joined Shomu's biology coaching for 6 month period of time.
So I'm really thankful for Shomu's biology, and I would suggest everyone to go for it.
A lot of help and support by Suman sir, always remembered in my mind.
The study materials I got helped me to understanding the topic very well, and the test series helped me to covering up the syllabus in a perfect time.
This is
And
all
also-
because of the online test series.
In-depth knowledge what Shomu's biology gave me. I want to thank Shomu biology. I want to thank each and every member of Shomu's biology for their effort, whatever they are putting on us.
And I am very much happy to choose Shomu sir for my CSIR NET GRF journey.
Really leveled up my preparation. I was confident on the day of exam.
The classes helped me a lot to understand my subject from very basics to advanced level.
So I really thanks Shomo sir and also ma'am for always guiding me.
With proper guidance, roadmap, and the help and training with perfect amount of study material and how to utilize them, the resources, everything together can help you qualify that. I am the person who will help you reach to the pinnacle of your success and your journey for the CSIR NET Life Science examination. If you trust me, if you trust my lectures, then join Shomu's Biology online coaching. Registration is going on. The details are there in the description. Fill the form to reach out to us or simply contact the number given in this video.